Oh my god, how am I gonna remember all this Ras, Raf, Gerb, and Gef?
I don't know how we're gonna do this.
Let's explain exactly how these students are going to remember all this Gerb and Gef stuff.
Memory exists in 3 different forms. The first is sensory memory, which only lasts a few seconds. This is the first time your brain will touch, feel, hear, taste, or smell this event. It will then become a short-term memory. In this stage, we can have short-term memory or working memory. The first type is for information that will be stored briefly and will often be repeated within the next couple of seconds, whereas working memory is information that you intend to manipulate and transform. To become a long-term memory, the information from the working memory must be converted. The hippocampus, a key part of the brain involved in memory, will collect this data and help it become a long-term memory. These types of memories can be further split up. Implicit memories are those you can remember unconsciously, like tying your shoes. Explicit memories require you to actively think about the memory, like the elements on the periodic table.
Speaking of the hippocampus is a really small seahorse-shaped structure located deep in the temporal lobe of the brain. Although it's small, the impact it has on memory is massive. Looking deeper, when information is sent to the hippocampus via the perforant pathway, it gets processed in specialized neurons called pyramidal neurons, located in the CA1 and CA3 regions of the hippocampus. When memory information first enters, it is through sensory areas in the cortex. A specific area in the cortex called the entorhinal cortex is the gateway to the hippocampus. If the memory constitutes working memory, the hippocampus works closely with the higher order part of the brain known as the prefrontal cortex. For emotional memory, the amygdala and other limbic system structures get involved. Lastly, for long-term storage, which we all know is never used when studying for our classes, the information is transferred to the outer part of the brain known as the neocortex.
The cells in our brain responsible for our thoughts, learning, and memory are called neurons. Neurons send electrical signals down their axons and communicate with other neurons using chemical messengers called neurotransmitters. Neuronal membranes are polarized, meaning they are more negative on the intracellular side of the membrane than on the outside. This is called the resting membrane potential, and it is maintained by sodium and potassium ion channels and pumps. Neurons receive signals from other neurons at their dendrites. When an excitatory signal reaches a neuron, a small patch of the membrane depolarizes, or gets less negative. If the signal is strong enough to reach the threshold, a rapid depolarization and repolarization of the membrane occurs. This is called an action potential, and it is the neuron's way of transporting a message. The action potential is propagated down the axon as it is regenerated by subsequent voltage-gated channels. Upon reaching the axon terminals, the action potential triggers an event such as the release of a neurotransmitter, which travels across the synapse to the dendrites of the postsynaptic neuron and triggers a response. This is the basis of how our neurons communicate.
Now, how exactly do neurons link back to memories? Memory is the reactivation of a particular set of neurons which is caused by the changes of strength of synapses. This process is called synaptic plasticity. The more you are exposed to a certain stimulus, the more active a certain connection is, and the stronger it becomes. These long-lasting increases in synaptic strength is known as long-term potentiation, or LTP for short. This is the key mechanism underlying learning and memory formation.
Now, let's explore the mechanism. Through the repeated stimulation of NMDA and AMPA by the neurotransmitter glutamate, magnesium is removed, allowing for glutamate to interact with NMDA. This allows for calcium to enter the cell, where it will interact with CAMK2. This allows for CAMK2 to activate itself, where it will activate NMDA, which will then activate AMPA, allowing for sodium to enter the cell and potassium to leave the cell, resulting in a net depolarization. Meanwhile, CAMK2 will also recruit more AMPA proteins to the cell membrane. This allows for even more sodium to enter the cell and more potassium to leave the cell.
As a result, we end up with a highly depolarized cell which activates many action potentials, increasing the efficiency of neural communication to and from brain regions involved in memory.
So, as you study or continuously rehearse information, this is what goes on in your brain. Let's do a quick recap of what we've learned to store this information in our own brains. Attending to and rehearsing information transfers it from working memory to long-term memory. This occurs by a process called long-term potentiation, the strengthening of particular synapses between neurons. This is particularly prominent in the hippocampus. Stimulation of a certain neuron leads to action potentials and the release of glutamate, the nervous system's main excitatory neurotransmitter. Glutamate binds to its receptors on the postsynaptic neuron and triggers a response. Through the activation of the CAMK2 protein, more glutamate receptors are delivered to the membrane. This sensitizes the postsynaptic neuron to glutamate, causing a stronger response each time it is activated. This is the neurobiology of memory and how we learn everything we need before exam day.
Oh my god, I forgot everything.